Welcome back to EASD TV. Now, earlier there was a very lively session indeed on big data and diabetes, and I've got three people who were involved in that session. So, introduce yourselves to us, please.
Sure. I'm Michael Snyder. I'm chair of the Department of Genetics at Stanford University School of Medicine.
Hi, I'm David James. I'm the chair of systems biology at the University of Sydney in Australia.
And I'm Romain Barrès. I'm a professor at the University of Copenhagen and Côte d'Azur in France.
What a wonderful affiliation, Côte d'Azur and Copenhagen. So, Michael, start us off. Big data and diabetes, where is this going?
Well, I think big data is going to permeate every field, including diabetes. It means sequencing people's genome to predict their risk for diabetes and catching certain forms of diabetes, which you can currently do, but it's not incorporated just yet. It also means collecting measurements on people at a level that's never been possible. And David will give you his analogy. I like to think of it as the way we do health now. You take a limited number of measurements, maybe 6 or 7, when we could take hundreds or thousands of measurements to get a much, much clearer picture of people's health and how disease comes on and understanding the basics of the disease.
Now, you are tooled up, if that's the right expression, with no less than 4 wearables. Presumably they are collecting data about you.
Correct. In fact, I usually use 8 or 9 of these every day. You're missing an obvious one, which is my hearing aids not only measure, not only assist me with hearing, but they also measure my physiology, heart rate variability, which all these things do. So they collect tons of useful data. Tons of data we're not sure how useful it is just yet. But we've shown that we can actually detect disease early using these devices. We can tell when you're getting COVID a median of 3 days prior to symptom onset with the simple smartwatch.
So we have Michael the evangelist. Tell us where you fit in, systems biologist.
Yeah, I guess I'm a little bit different to Michael in the sense that we're trying to use systems biology to understand basic mechanisms of complex problems. Most of the diseases like diabetes that we work on are very complicated. And what we've really come out of over the last 3 decades or so is a landscape where people were very reductionist. They were focusing on individual molecules, thinking that these were the holy grails, when in fact we now know that the answer is many things. And unless we embrace the complex landscape of the whole system, I think we're going to fail again and again and again. And we see this with clinical trials. The number of drugs that go up and fail is just too many, and it's costing us too much money. And I think the real benefit of what we're talking about is if we can hone in on key regulatory molecules that are driving the whole system, they're much more likely to be the best drug targets and succeed in the clinic.
So you're using big data to try and pinpoint the right pathways
Correct.
to the druggable targets.
Exactly.
Romain, how are you using big data?
Well, I work on epigenetics, and recently epigenetic has been used as a tool to make sense of genetic data that we gathered for many decades. So the genome-wide association data have identified that if you have a mutation somewhere, you're more at risk of a certain disease or being obese or even insulin resistant. What we could do now using these omics related to epigenetics is to make sense of these mutations and put them in the context of the real cell, where DNA is not a linear molecule with an alphabet that goes very linear, but it does interact and fold and form 3-dimensional structures. And epigenetic signals that we detect through these omics that we use in my lab are allowing us to know which mutation interact with which other region of the genome in time and space. When we exercise, is this region relaxing or condensing, etc. So without these methods, we wouldn't make sense of these genetic data that we gathered a long time ago.
Now, on behalf of all the diabetologists watching who may be thinking, oh my goodness me, what are they talking about? Omics and epigenetics. Epigenetics, I guess you could say, are like post-it notes that go on the genome that, make more of this, make less of this, don't turn this one on, until tomorrow tea time, that kind of stuff. Explain omics for people who don't or not familiar with that word.
Okay, I like to use an analogy which is sort of close to home. If you think of a drunk person, and this is a story many of us.
This is not to say that he's drunk all the time just because he's an Australian.
Thank you very much for that.
I just want to make that point.
Yes, bit typecast, But the Australian drunk person is walking home from the pub and he's scratching around on the footpath, and a policeman comes along and says, "What are you looking for, sir?" And he said, "I've lost my keys." And the policeman says to him, "Oh, did you lose them here?" He said, No, I lost them over there in the darkness." He said, "Well, what are you looking for them over here for?" He says, "Because this is where the light is." And this is the whole point of omics. For too long, all of us have been focusing our attention under the lamppost. And yet out there in the darkness is a vast land of opportunity. That could be where the secrets lie.
So basically, it's the downstream consequences of what happens when genes are turned on and off.
Yeah, that's one way of looking at it, yeah. But I mean, there are many things that we have to look at. I mean, if you think of it, Romain talked about epigenetics, and that's sort of a long-term program. It turns things on and off over a scale of hours. But actually, the biology plays out, in many cases, over a period of minutes. And so we've got to be able to measure omics at multiple levels that captures all of this. As we're eating food, as we get an infection, you have to stress the system in order to see how the system is set up. And that's really the goal of omics.
But this all generates an extraordinary quantity of data. How do we sort out the wheat from the chaff? How do we make sense of all that data?
Right. Well, right now, a lot of emphasis is on collecting the data of all forms. And I want to emphasize something David said. We're really collecting all kinds of forms of genetic data that's downstream. That's all the way to the metabolites and things like that. And that's what all of us are doing. And we're also doing it at all levels. I think David and Romain are a little bit more focused on upstream things. What's really going on in diabetes? What are the early events? We're actually more applied. How do you use that to try and understand when things go off? So I think we're trying to look at all these levels, and now I've forgotten your question because I got diverted.
But it's how do we? And I'm looking at you with all your
we need visualization tools.
Yeah.
It's like a rocket ship or even your car. Your car has 400 sensors on it, but you don't get information about 400 sensors back. It would fry your brain if you did. Instead, you have a dashboard that integrates all this information into form that makes sense that you as a person can use, and that's what we need to do it for physicians. Basically, take all this complex data, figure out what parts are really useful, and bring the useful parts back in a form that people can use. And in fact, I believe you should bring it back to the form the patients can use, not just the physicians, because patients have more time to spend thinking about themselves than a physician does. So if we can bring it back into the form that everybody can use, we'll all benefit.
And presumably, AI is very important in this because you've got so much data, you really need AI to trawl through it.
Yeah, you're raising a really important issue that all three of us deal with, which is the personalization side. We're all different. And big data lets us follow that. That is to say, the way you'll respond to eating a banana will be very different to the way I respond to eating a banana. And some people's glucose, especially relevant to diabetes, some people's glucose goes out of whack after they eat pasta. For other people, they're perfectly fine. But those who are fine for pasta may go off with this banana or rice or other things. So we all respond to food differently. We all respond to environmental stimuli differently. And big data and omics lets us see that picture and actually modify our behavior as a consequence. And these wearable devices, they're actually glucose monitoring systems that are extremely powerful for doing all this. So all that can be brought in with AI in a personalized fashion.
But what do you say if the black one says one thing and the white one says another thing?
Well, believe it or not, they tend to agree when they're fairly accurate.
Few.
No, I think like heart rate variability, they're all pretty similar. Now, they will differ on your skin temperature and other things, but actually the glucose monitors are pretty accurate, it turns out, even across different monitors. But I think where it gets wacky, maybe this is what you're getting at, is one form of data may tell you a different picture than the other, and that's because we honestly don't understand yet how to put those together. And that's where the basic science comes in. We have to bring all these data types so we can get a much more detailed understanding of how complex biology works. And David's a master at this.
So how does the basic science from you two flow down to folks like Michael?
Well, we work together. We work off each other, And I think the important aspect of this, and I think Michael touched on this, it's critical. We're now in a collection phase. We're out in the jungle collecting all the bits and pieces that make up the puzzle. But critical now is to make all of that publicly available, okay? And then I think the next level is to engage people who are expert in analysing data. And this is where the big challenge is. And we're about to move to a time, some would argue we're already at the time, when people don't do experiments anymore. They sit behind a computer, they access UK Biobank and so on and so forth, and can generate reams of papers and information, and actually, in some cases, drugs that can be used in the clinic.
You're nodding your head there. Is that where you see things going too?
No, I think it's an interesting. I don't have this vision. I think there'll still be a need for a physiologist to test and to do in vivo experiment. But I like the idea, and there's actually a lot of data that is sitting there and that's not sufficiently analyzed because we are lacking resources. And AI can help us, but it needs to be supervised by human beings. So I think in this new era that we're living, I don't know if Mike and David will agree on this, but we need more people that are computational biologists that understand biology in order to make use of data, because we have accumulated a lot of data already, and there's a lot of it is sitting almost still in databases, and so we need people to generate hypotheses based on this data. We need computational biologists, and eventually we need to validate the targets, the drug, there'll always be a need of physicians and there'll always be a need of physiologists to understand the system.
Can I just say something to the audience? If you want to be kept in your old age by your children, get them to be computational biologists. There is a worldwide shortage of them. They are paid squillions, the best of them, and they will always be in a job.
100%.
Just to be serious a moment, I mean, Leroy Hood, the guru of systems biology, has been talking about this for, the hype of the 4 Ps of precision medicine, which is where this is all going, of course, precision medicine. He's been beating the drum for this and systems biology for the past 25, 30 years. Are we over the hype and into the delivery?
Yeah, I mean, I think people say that to us all the time. We get that. People say, well, you guys have been trumping on about this and we've seen nothing. But the truth of it is, this is a big ambition. And these things don't happen overnight. As Romain just said, we've still got all these isolated data sets sitting around, and what we now need to do is build all of these together in an integrated way and make it accessible to the world. And that's when I think all of the fruits of our labours will start to come to fruition. We're seeing that now. And, Mike, for example, he published a paper in Cell, the best journal in the world, on himself, where he collected, you should ask him, it was a fascinating study. He collected lots and lots of data in himself, and he actually diagnosed himself with type 2 diabetes using Omics data. Right.
To get back to your point, 25 years ago, it was just a dream. It's only at the beginning, I would argue now. We have to figure out, we're collecting data, we're trying to figure out what parts are useful. Everybody knows your genome sequence is useful, and it's only now we're starting to be able to interpret that, to actually bring it into the clinic.
But your genome is not your destiny, and just because you have variants that might increase your risk of type 2 doesn't mean that you will get type 2
agree.
diabetes.
It's true for breast cancer, right? If you have a BRCA mutation, you're a 50% chance of getting breast or ovarian cancer if you're a woman. You're not 100% chance, but that's still very useful information to know, right? You'll get checked up a lot more. And that's the way this is all going to play out. We're going to get probability scores for all this risk prediction. Some we're much better at now than we were even 5 years ago, much, much better. And we're still a long ways from predicting a lot of complex diseases. But we will get there. And you're right, you need the other information, but imagine this. We can now take certain things like your genome sequence, add in some of your electronic health information like LDL, LDL and cholesterol levels, I should say, bad cholesterol and good cholesterol. And they're both important. You can actually mix this in, and with machine learning and AI, make better predictive scores. This is where we're going. We're still at the very, very early days, and we're just starting to show this, but we have to show utility. We have to show the stuff does work and has information. That's how we're going to convince physicians. 10 years ago, when we went around sequencing healthy people's genomes, physicians were mad at me. They said, all you're going to do is turn everybody into hypochondriacs. This is a terrible thing to do. They don't say that so much anymore. And I think that's how it's going to work. And people still tell me now with big data, this wearable stuff, you're doing a bad thing. You're giving people all this information. You just turn everybody into hypochondriacs. But now suddenly we're starting to pick up infectious disease from a smartwatch. Well, maybe that's not so bad after all. Maybe that does have utility. And I think that's how we're going to transform medicine. Show it's useful, then figure out who's going to pay for it. That's a big part,
and
too.
And the final bit, which is really important, that you develop the APIs, the kind of electronic handshakes that allow this information to be integrated into clinical records.
100% agree.
And that people then, doctors intervene when they should be intervening.
Along with that, we have to educate physicians that this is important and this is useful. The instant reaction when you tell a physician, like genome sequencing 10 years ago, like everybody, here's a good example now, everybody knows the microbiome's important, yet nobody's shown utility, and that's why it hasn't found its way. As soon as we show utility with the microbiome, it will find its way into the electronic health record. And we'll have to educate physicians, just like we have to do now about genome and wearables, that these are important, useful pieces of information, and they can help them in their clinical care.
It's a fabulous session. It was a fabulous session. And of course, the great thing is for you that you can look at it on the EASD web service if you missed it. And if you did miss it, I really urge you to go have a look at it. Thank you all so much. Evangelists, all of them. Bye for now.